Illnesses, especially rare and chronic diseases, often evoke feelings of indescribable fear and helplessness. We need more research, more allies, and more advocates, and I consider myself to be a soldier in this fight. Welcome to Chronic Illness and Advocacy. My name is Lakshmi, and today we have Dr. Celeste Simon, an esteemed researcher at the University of Pennsylvania. As the scientific director of the Abramson Family Cancer Research Institute and co-director of the Parker Institute for Cancer Immunotherapy, she focuses on how cells respond to low oxygen levels and how this affects tumor growth. With numerous accolades, we're excited to learn from her insights in cancer research. Thank you for joining me today.
My pleasure. Happy to be here.
So, can you tell us about your background and how you became interested in studying cancer and immunology?
Well, I've been at it for a long time. I got interested in science taking a chemistry class in high school. Went to college with the plan of majoring in chemistry, but switched to microbiology. Began getting interested in cancer towards the end of my bachelor's degree and started studying RNA tumor viruses as a master's degree student, and then finally as a PhD student at Rockefeller University. I sort of pivoted away from viruses for a few years, but got back to really focusing on cancer when I was recruited to the University of Pennsylvania in 1999 to help initiate the Abramson Family Cancer Research Institute. We started because of a $100 million gift from the Abramson family that wanted to have a major impact on Penn and how it both delivered cancer care, supported cancer survivors, found new mechanisms underlying cancer cause and treatment. And I would say it's been going strong for 23 years now.
So your research focuses on the tumor microenvironment and how it impacts cancer progression. So what are some of the key findings from your work in this area?
Well, when I was a PhD student, we thought of a solid tumor as basically a collection of malignant cells. But in fact, solid tumors are a multicellular complex ecosystem where the cancer cells, which are already very genetically diverse, are infiltrated by very diverse heterogeneous populations of both immune cells and what we call cancer-associated fibroblasts. They're invaded by blood vessels. They're actually innervated, and neurites and axons are delivering things into the tumor microenvironment. And so the disease progresses and metastasizes because of all of these cellular constituents. And I think the complexity underscores why this has been such a difficult disease to treat. You're not only fighting the cancer cells, but for example, the recruited immune cells or the recruited fibroblasts can be both tumor promoting and tumor restraining. So you have to figure out the good guys versus the bad guys. And in tandem with the cancer cells themselves, you want to eliminate the cancer cells, eliminate the cells in, the support system that drive tumor growth, and enhance the activity of those that will kill off the tumor and hopefully avoid metastasis. At this time, metastasis is really why most cancer patients die. A lot of progress is being made on primary tumor control, but it's the metastases that remain a major, impediment to overall curing people. So I think that's why studying all the cellular constituents, if you will, of the tumor microenvironment is so incredibly important.
So how do you think this knowledge can be translated into new cancer treatments?
Well, already the knowledge is having a major impact. I think one of the stars would be T lymphocytes, T cells, right? So many forms of immunotherapy are specifically directed at, and the T cells are the same thing. We can have good T cells that get rid of the tumor, and we can have bad T cells that impact the functionality of the good T cells. So, the immune checkpoint approach is meant to rev up the good T cells at the expense of the bad T cells. The adoptive T cell therapies like CAR T cells or T cells with transgenic T cell receptors. Again, how do we enable their functionality, their persistence, their return if the cancer returns? So just by understanding how complicated that is has gotten us to, the immunotherapy approaches that are becoming in more and more widespread application. But now people are really beginning to look at the other players. They're trying to come up with chimeric antigen receptor modified macrophages. They're trying to thwart the activity of what we call suppressor neutrophils that are, again, decreasing the functionality of the good T cells. People are looking at the fibroblasts. How are they supporting the tumor? And a really hot area is how is the nervous system supporting the tumor? That's a very new area. And so a lot of work is really just trying to parse all this out. It's incredibly complicated and it varies patient to patient. But the more you know, the better you'll be at, specifically engaging the good players at the expense of the bad players. And where things have gone horribly wrong is when we thought, for example, all T cells were the same or all fibroblasts were the same, and they're not. And so, you really need to understand who's who and engage the ones you want and, hopefully eliminate the bad people. And, along with eliminating the cancer cells themselves. So it's very tricky, but I would say the payoff at this point for really understanding the tumor microenvironment is definitely the immunotherapy approaches that are now, in some cases, being effective. Another area, probably the first players to be dissected were the blood vessels. You probably heard of anti-angiogenic therapy. This was very exciting in the 1990s, and it didn't live up to hopes. In fact, though, the anti-angiogenics actually do work for certain cancers, and quite frequently now they're being combined with the immunotherapy approaches, because if you can change the quality of the blood vessels, the idea is the good T cells will get there more rapidly. The other idea is if you change the quality of the blood vessels and the lymphatic vessels, maybe you'll avoid metastasis because that's how they escape. That's how the metastatic tumor cells escape. So I think the other thing is we should remember that things go in cycles. So in the 1990s, people were very excited about anti-angiogenics. That died down, but now years later, people are finding ways to repurpose those approaches and similarly, in the 1990s and early 2000s, while people were very excited about anti-angiogenic therapy, they thought immunotherapy was going nowhere. And people like my colleague Carl June never gave up and kept plowing away, and here we are. So it has very cyclical applications, I would say, as we learn more.
Yeah. So you received numerous awards for your research, including the prestigious Lasker DeBakey Clinical Medical Research Award. So,
actually, I need to stop you there. I actually did not. I think you got that idea because three very good friends of mine did.
Okay.
I work in the hypoxia research area, and it was in 2016 that three very close friends, Peter Ratcliffe, Bill Kalin, and Greg Semenza, won that award. Then 3 years later, in 2019, they won the Nobel for their important contributions to the field I've been working in for over 20 years. While I didn't win either of those awards, I feel like I was very connected to their efforts to the point that I actually got invited to the Nobel ceremony and banquet as a guest of the Nobel Committee. That was enormously exciting. And I would also say that we've laid the foundation for a new FDA-approved drug for certain kinds of cancer, the HIF-2 alpha inhibitor. So that's the reward I've gotten for what we've been doing. But I wrote essays about the Lasker Award, but I myself did not win it. So just wanted to make sure That's fine. When I saw that question, I'm like, oops, I better correct it. I have one other rewards, which I greatly appreciate, but that was not one of them. But I wrote essays for the Lasker Award when they won. I wrote essays for the Nobel when they won. The day the Nobel was announced, October 7th, 2019, I spent roughly 48 hours speaking to reporters from the New York Times, the Wall Street Journal, Science, Nature, Philadelphia Inquirer, on and on and on. So I feel like I was very connected to what they did, but I did not win either the Nobel or the Lasker, but that's totally fine. I didn't go into science to have that happen. So yeah, just to correct
So,
the record.
yeah, so in general, like, you have won, like, several other awards. So
Mm-hmm.
what exactly are the significance of awards in terms of impact it has had on your work?
Well, we go into research for a variety of reasons. Many people go into research just because they're curious. They just want to understand how the world works. If you figure something out, it's very gratifying. If you work in cancer biology, you're primarily in that area because you want to address this terrible clinical problem we face. But in parallel, you always learn something new about normal cell processes or tissues. And that's why we go into it. You have to work very hard. You have to learn to overcome big setbacks in your research, rejected grants, rejected papers, frustrations. It's a very rewarding field, but you survive the field because you learn to be happy with the successes and cope with the many setbacks, and everybody has them. On the other hand, occasionally you do win an award, and that's very, that just shows people appreciate your effort. So I guess a couple that would mean the most to me is I was appointed a Howard Hughes Medical Institute investigator for 20 years in 1994, which meant we could do very courageous things with our research. That was really appreciated. Since that time, I've won a FASEB award for mentorship, which means a great deal to me. I've just won an award from the American Association for Cancer Research that I understand is prestigious. So those things, obviously, they're a shot in the arm, and you feel like people recognize your effort for. I started really doing this in a very serious way in 1977. When I first entered a master's degree program, and all these many years later to see that some things are paying off, like the HIF2 inhibitor. That's very gratifying. But the occasional award is a shot in the arm, and sometimes they come on a day when, something bad happened in the lab or some paper got rejected, and it just kind of helps buoy you and keep you going, because like all jobs, you have good days and bad days. And
Yeah.
those rewards help you get over the bad days, I guess.
So you've been a mentor to many young scientists over the course of your career. So could
Mm-hmm.
you discuss the importance of mentorship in science and how you approach mentoring your trainees?
So it's hugely important. I think, and mentorship is changing, I think, for the better. When I did my PhD at Rockefeller University, their philosophy was, it was on you. They would provide the laboratory, the wherewithal, but you were much more of an independent operator, there. It was very hard. But the philosophy back then was that if you could be that independent and a self-starter, that would make you more likely to succeed, and many people did. Not everybody. Some people gave up. It was pretty hard. Now expectations are, and I think this is a good thing, that mentors have a huge responsibility. And so when I became a mentor in 1992 with my first faculty position, I really wanted to approach that with the idea that it was a huge responsibility, that I wanted people in my laboratory to have a different experience than maybe some of the labs that I worked in. Not that they were bad or the mentors were bad, but I really kind of wanted to be a different kind of mentor. And I just think that, sure, I made a million mistakes, but I'd like to think that that's helped. I think I've had, if I really add everybody up, over 150 people through the lab over the years. I've had roughly 40 PhD students and about that many postdoctoral fellows. Over above that, we've had research technicians, lab managers, high school students, undergraduate students, medical fellows, et cetera. I think it's been really great just to see how many of them have gone on to be hugely successful in their own right. That's because they're very talented. But, getting a lot of input, a lot of advice, I think increases the likelihood of their success. That's probably the most gratifying thing that's happened, is to see people that have come through the lab go on to be successful in their own right. You have two principal goals. Goal number one is to cure cancer. Goal number two is to train the next generation of cancer researchers. And frankly, they're both equally important in my mind.
Right. Yeah. What advice do you have for young scientists who are interested in pursuing a career in cancer research and immunology?
Well, my advice is, get into a PhD program. It's a great time to be a PhD student right now. First of all, we've had a big expansion in biotech, biopharma. When I was a PhD student, if you wanted to go into industry, they still wanted at least 3 years of postdoctoral training. The expansion has meant that many of our PhD students, if they want to go to the industry route, will go directly into industry now. What that means, though, is that now there's a shortage of academic postdocs. So many labs, some of our very top labs, are looking for postdoctoral fellows for the first time in their career, which means if you're a PhD student that wants to go to an absolutely top-notch lab, the likelihood of getting in there is terrific. Moreover, you can use your PhD to go into public policy. You can go into not-for-profit. You can go into consulting. So this is just a great time to be a PhD student, and it can be a jumping off point you could go into patent law. Yeah, there's so many directions one can take with their PhD. Pick the best possible program, pick something you're passionate about, pick a good mentor. And, there's so many directions you can take. At the end of the day, everything's interesting and exciting, but I think students and everybody really try to pick something they're particularly excited about. Cause it's hard. It's long hours. It's demanding. It can be frustrating, but, if you persist, things work out.
And then last but not least, looking to the future, what are some of the most promising directions in cancer research, and how might your work contribute to these efforts?
Well, so I think everybody would say right now we're in the midst of an immunology revolution. And in fact, I think that extends beyond immunotherapy for cancer. These things could now be harnessed for autoimmune approaches, other, cardiovascular diseases. So there's that effort. I'm proud to say that the mRNA vaccines were developed at Penn, and we have a new RNA Technology Institute and how you can use RNA molecules to, treat, again, immune disorders, cancers, pathogens, cardiovascular issues. That's a very rapidly expanding area based on the, enormous success of the mRNA vaccines for the COVID response. All along, targeted therapies have been very important. And I think now we have more and more ways to drug what we used to call the undruggable targets. So the HIF2 alpha target for kidney cancer was deemed always to be, quote, undruggable. Well, it's been drugged, so I would say I think the next decade is going to see, and I'm focusing on cancer per se right now, not to mention, all the other diseases that people contend with, but for cancer, I think we'll see, immunotherapy approaches expand and be refined. I think we'll have more and more undruggable targets get drugged. I think that RNA technology is going to be pretty spectacular. I think that new forms of radiotherapy. So Penn, 2 decades ago, invested in proton therapy to deliver precision radiotherapy for especially pediatric patients. So the problem with radiotherapy is it works. The side effects, though, are you can, be toxic to normal tissues. So proton therapy and so-called flash therapy are supposed to make the radiotherapy, the radiation treatment much more precise and spare normal tissues because, people can be cured of their cancer, but then have, long-lasting toxicities that can really diminish their quality of life as a cancer survivor. We're really trying to address massive healthcare disparities. Those are very important. And also, every walk of medicine is going to have a component of treating cancer survivors. As more and more people survive, if you're a pulmonologist or you're a cardiologist or you're a dermatologist, you're going to be treating cancer survivors. And that means we're having some success, but it means there's new challenges in, the biomedical enterprise. So that's how I would answer that question for cancer, but that's just one of many illnesses that people contend with now.
Yeah, and I know that I said that last question was the last question, but I have a last minute question for you. So I've just recently learned about CRISPR.
Mm-hmm.
So are there like cancer eliminating, I guess, therapies or like?
Right. So I should have mentioned CRISPR as a major new development. Of course it is. So, people really think eventually we could get to the point where genome editing could mean you don't have germline mutations that make you predisposed to getting certain forms of cancer. I would say those ideas are a bit dangerous right now. Where CRISPR is certainly being deployed, though, is, for example, they're now using CRISPR to make, for example, CAR T cells better at killing a cancer, persisting, and coming back, when the cancer reappears. So that's already happening. People are using CRISPR to look for, let's say a cell doesn't respond to a therapy. You could do a CRISPR screen to find an additional target. If the two were combined, then the cells would die. So that's another big area. That's just a few examples, but you're absolutely right. Along with RNA technology, CRISPR technology is, an amazingly rapidly expanding area that the technology just changes daily. It's sort of amazing. So, I think we probably don't even have any idea how this, ultimately is going to affect everything we're doing. So,
yeah. And I apologize for the mix-up with the last Great Debate Award again.
No worries. I mean, geez, I wish I did, but that's all right. I am very friendly with people that did, so that's pretty cool.
And so it's always a reward to hear experts like yourself speak. And what you had to say was really valuable. And thank you again so much for your time.
You're very welcome.